Welcome to the deep dive. This is where we take a stack of complex information, sift through it all, and really pull out the most important bits for you. Today, we're diving into the microscopic world. It's, often overlooked, but it's just teeming with incredible life doing really crucial things. When we think fungi, yeah, sure, mushrooms come to mind. But what about those tiny hidden guys in the water and soil? The ones working behind the scenes?
That's exactly right. They're not just curiosities. They have a huge impact.
Oh,
Unseen architects, maybe. Recyclers.
exactly. So our mission today is to unpack a really fascinating, maybe a bit peculiar group, the phylum Hyphochytriomycota. We're using a chapter from an introductory mycology text, and honestly, there are some surprises in here about these tiny things that kind of challenge how we classify life.
Yeah, Hyphochytriomycota.
Yeah.
It might sound like just another obscure name on a list. But what's really cool is how they show the sheer diversity within what we loosely call fungal-like organisms. They're a small group, definitely distinct. Historically, people got them mixed up with true fungi, but they've got unique features that really set them apart. It makes us constantly refine how we see the tree of life.
So, why should you, listening right now, care about these microscopic creatures? Well, this deep dive, it's your shortcut. You get the core understanding of a complex topic without wading through, all the dense textbook stuff. It's about seeing the incredible variety of life out there and how scientists are always figuring out these tiny but mighty players in our ecosystems. Okay, let's unpack this. Prepare to meet some of biology's most interesting, maybe most misunderstood microscopic residents.
So first things first, what are Hyphochytriomycota? Well, fundamentally, they live in soil or water. And for a long time, like I said, they were sort of lumped in with chytrids, another group with flagellated cells because, well, they looked a bit similar on the surface. But now, with better genetics, better microscopy, we know they're actually only very distantly related to chytrids.
It really shows how science evolves, doesn't it?
And here's the really distinctive part, their defining feature. They have these motile cells, zoospores. Each one has a single tail, a flagellum right at the front, the anterior end. But it's not just a smooth whip. Imagine like a tiny swimmer propelled by this tail. But the tail itself is covered in these fine microscopic hairs all along its length, almost like a tiny bottle brush. This hairy flagellum, that's the key identifier, right? Sets them apart. Absolutely, that's a great visual. Those flagellar hairs are their signature, no doubt. And ecologically, they're pretty versatile. You find them acting as parasites, often on algae or other fungi, or they live as saprobes, meaning they feed on decaying stuff like dead plants or insect bits. The marine ones specifically tend to parasitize marine algae or sometimes aquatic animals. And it's funny, despite these roles, it's a really small group, only about 23 known species. But even in that small number, there's quite a bit of variation in how they're actually built.
That small number is surprising. So how are they built? What does their body, the thallus you called it, what does that look like?
Right? The thallus, their vegetative body. It does show some interesting parallels with chytrids, which maybe hints at, some deep evolutionary connection way back. Generally, in the Hyphochytriomycota, we see two main types of thallus structure. First, you've got the holocarpic species. In these, the entire organism develops inside its host, its endobiotic, and the whole thing, the entire thallus, converts directly into a zoosporangium. That's the structure that makes the spores. A good example is Anisolpidium ectocarpii. It lives inside a marine brown alga. The whole organism just becomes a sack of spores. Very efficient, you could say.
The whole thing. Wow. Okay, so that's holocarpic. What's the other type?
The other main type is eucarpic. Here, only part of the thallus turns into a reproductive structure. The rest, the vegetative part, stays intact. And within the eucarpic forms, there are a couple of variations. Some are monocentric. That means the thallus is basically one main reproductive part, but it has this branched root-like system attached called rhizoids. Think of it like a central hub with these little anchors spreading out to grab nutrients. Rhizidiomyces apophysatus is a classic monocentric example.
Okay, a central hub with roots, and the other eucarpic kind.
That would be polycentric. These are more complex. The thallus is made of branched filaments called hyphae, and these often have internal cross walls or septa, so it forms more of a spreading network, kind of like a mini root system weaving through whatever it's growing on. Hyphochytrium catenoids is like this. And here's a really interesting physiological point, something that applies to all the species studied so far. Their cell walls contain both chitin and cellulose.
Both. Chitin and cellulose, isn't that unusual?
It is. Very unusual. Chitin is typical of fungi. Cellulose is typical of plants and some protists. Finding both together is rare, and it really makes biologists think about those ancient evolutionary lines. It sort of blurs the boundaries we thought were so clear between kingdoms. Life's just more complex than our neat boxes sometimes allow,
That's incredible. A real biological curveball. Okay, so with these structures, how do they actually multiply? How do they spread? What's their typical reproductive cycle?
Good question. And actually, their asexual cycles are pretty similar across the board, regardless of whether they're holocarpic or eucarpic. It always starts with those unique zoospores, the ones with the hairy flagellum. After swimming around for a bit, they find a good spot, a suitable substrate or a host, and they insist. They basically settle down, pull in their flagellum, and form a protective cyst. Now, from that cyst, one of two things happens. Either it develops directly into a new thallus right there, or it first penetrates a host cell. If it penetrates a host, it then kind of empties its content as protoplast inside the host cell, and then the thallus develops inside.
Okay, got it. So it either develops outside or injects itself inside first. Then what?
Then the mature thallus, whether it developed inside or outside, eventually gives rise to or gets converted into a zoosporangium. And these are inoperculate zoosporangia. That just means they don't have a little lid or cap that pops open. Instead, new zoospores with that single anterior hairy flagellum are released through special little tubes called discharge tubes. And off they swim to start the cycle again. And again, you'd be looking for those distinctive flagellar hairs under the microscope to be sure it's a hyphochytrid zoospore. There's also a cool little detail inside the zoospore, a second kinetosome, like a basal body structure. Its exact role isn't fully clear, but it's there.
Right, so that asexual cycle seems pretty well mapped out. But what about sex? Does sexual reproduction happen in these organisms? Or are they strictly asexual?
That's a key point, and honestly, it's one of the big unanswered questions for this group. As things stand, sexual reproduction has not been conclusively proven in any hyphochytrid.
Really? Not at all. That seems quite surprising for a whole group. But you said conclusively. Does that mean there are hints? Or just nothing?
You picked up on that nuance. Yes, there has been some evidence reported that suggests a sexual cycle, specifically for Anisolpidium ectocarpii. Remember, that's the holocarpic one living inside the brown alga ectocarpus. In that specific case, researchers observed adjacent phalli inside the alga actually fusing together. And then they saw their nuclei fuse. That's called karyogamy. The nucleus that resulted from this fusion, the suspected zygote nucleus, then divided multiple times. And the wall around this structure thickened up considerably, forming a really tough, thick-walled resting structure.
Okay, so fusion thick wall sounds promising.
It does. And the idea, the postulation, was that this resting structure could survive harsh conditions, maybe overwinter, and then later germinate to release new zoospores. And this is the crucial, but meiosis has never been directly observed. Meiosis is that special type of cell division that shuffles genes and reduces chromosome number, and it's really the definitive proof of a standard sexual cycle. Without seeing meiosis happen in Anisolpidium or any other hyphochytrid, we can't definitively say they have sexual reproduction in the traditional sense.
Ah, I see. So suggestive signs, but missing that final critical step for confirmation. It really shows how rigorous scientific proof needs to be, doesn't it?
Exactly. It highlights that even strong circumstantial evidence isn't enough sometimes.
Yeah.
You need to observe the key processes. And maybe their reproduction just works differently than our standard models. It opens up questions about life's diverse strategies.
That makes sense. Okay, so with all these unique features, the hairy flagellum, the chitin cellulose walls, this mystery around sexual reproduction, where do they actually fit in the grand scheme of things? Where are the Hyphochytriomycota on the evolutionary tree?
That's a great question, and it really speaks to how our understanding changes, especially with new tools. As we mentioned, they looked a bit like some chytrids, so much so that for a long time, the few known species were just put in with the chytrids in the order Chytridialis. This was despite that really important difference in the flagellum being hairy and at the front, but maybe it was overlooked or considered less important initially. As more species turned up, though, and scientists looked closer at these details, it became clear they were fundamentally different. They really needed their own group, separate from the true chytrids.
So where did they land eventually?
Well, current classification puts them in their own phylum, Hyphochytriomycota. And interestingly, this phylum is often placed within the kingdom Protoctista, or sometimes grouped with other Strematopiles. And here's where genetics becomes super important. The hypothesis now, which has strong support, is that their ancestors were actually heterokont algae.
Algae. Really? How did they figure that out?
It comes down to similarities in the flagellar structure. Heterokont means different flagella. And while hyphochytrids only have one visible, the internal structure and the presence of that second kinetosome are suggestive. But the really strong evidence comes from DNA sequence analysis. Comparing their genes to other groups points quite clearly towards an origin within or alongside these heterokont algae, which include things like diatoms and brown algae. So modern genetics connected dots that morphology alone couldn't quite solidify. It really shows how these organisms, once kind of shoved into the wrong box, are helping us redraw that tree of life based on deeper relationships.
Fascinating. A link to algae confirmed by DNA.
Okay, and within this phylum Hyphochytriomycota, how is it broken down? Are there lots of smaller groups?
Actually, no, it's quite streamlined. The entire phylum consists of just one order, the Hyphochytrials. And within that single order, there are only three families currently recognized: the Anisolpidiaceae, the Rhizidiomyces, and the Hyphochytriaceae. And these families are distinguished mainly by that phallus morphology we talked about earlier.
Right, let me see if I remember. Anisolpidiaceae are the holocarpic ones, single cell, no rhizoids, like Anisolpidium.
Exactly. Then the Rhizidiomyces are the eucarpic, monocentric ones, single reproductive structure with rhizoids, like Rhizidiomyces.
Okay, and the Hyphochytriaceae must be the polycentric ones with the branching hyphae network, like Hyphochytrium.
You got it. And the biggest of these families is Rhizidiomyces. It includes Rhizidiomyces apophysatus, which is probably one of the most studied species. We could maybe walk through its life cycle in a bit more detail. Might help make all this feel more concrete. Yes, please. Let's zoom in on Rhizidiomyces apophysatus. Give us the play-by-play. What does its life actually look like?
Okay, Rhizidiomyces apophysatus is pretty cosmopolitan, found all over the world. Europe, the Americas, Asia, Africa. You often find it as a parasite on water molds, like Saprobic species, or on the alga Vicoria. But you can also isolate it from soil samples or even pine pollen floating in water. Shows it can handle different environments. And remember, its thallus is monocentric, so basically a single cell body with rhizoids. And that whole cell body eventually becomes the zoosporangium.
Got it. So start with the zoospore.
Right. Starts with that tiny zoospore swimming actively with its single anterior hairy flagellum. It swims around for, say, 25 minutes up to maybe 90 minutes. When it finds a suitable spot or maybe bumps into a host, it encysts, rounds up, pulls in the flagellum, forms that protective cyst wall, and pretty soon after encysting, the nucleus inside starts dividing.
Okay, it's settled down. Then what happens?
As the organism inside the cyst starts to grow, it puts out a little germ tube, a primary rhizoid, which penetrates the host cell wall if it's a parasite. And here's that interesting feature. Often, right where that rhizoid goes into the host, a swelling forms on the rhizoid just inside the host wall. The swelling gets bigger and becomes what we call the apophysis. It means outgrowth or offshoot. It's considered part of the developing sporangium. The main part of the sporangium develops from the original encysted zoospore body sitting on the host surface. So you've got the main body outside and this apophysis bulge just inside the host connected by the rhizoid.
Okay, I'm picturing it. Main spore sac outside, little bulge inside the host wall. Connected.
Exactly. Now, as the sporangium matures and gets ready to release the next generation of zoospores, a clear spot, a little bump called a papilla, forms on its surface. Then, from this papilla, a tube starts to grow outwards, the discharge tube, like a little spout forming. The cellular contents, the protoplasm, which is now multinucleate from those earlier divisions, flows into this discharge tube. Then the very tip of this discharge tube swells up, inflating into a thin-walled bubble called a vesicle.
A bubble outside the main sporangium.
Yes, outside. The protoplasm moves out into this vesicle, and then inside this external vesicle, the protoplasm finally divides up, cleaves, into individual unenucleate zoospores. Each one develops its new hairy flagellum, and then pop. The vesicle bursts, releasing all these new zoospores, which swim away to find new hosts or substrates, and the cycle begins again.
Wow. So the final separation into individual spores happens outside the main body in that little balloon. That's intricate.
It really is a fascinating variation on spore release.
Okay, that detailed look really helps bring it to life. So pulling back again to the big picture, why does all this matter? Why should we, as learners, really care about these 23 species of tiny, hidden organisms? What's their real-world relevance?
Yeah, it's a fair question. Why focus on such a small group? Well, first, ecologically, they do have an impact. As decomposers, the saprobic ones, they're breaking down dead organic stuff, recycling nutrients in soil and water. That's crucial. And as parasites, they're helping to control populations of other microbes, like algae and various water molds or even other fungi. They're part of that complex food web, that system of checks and balances in microscopic ecosystems. They're the unseen cleanup crew and population managers.
So, important for ecosystem function, even if unseen. What else?
Well, from a research perspective, they're actually pretty useful tools. The fact that you can isolate some of them, like Hyphochytrium catenoids, and grow them in the lab, even on defined chemical media, that's really valuable. It means scientists can study fundamental biological processes in a controlled way. Things like How do they build that weird chitin cellulose wall? How does that hairy flagellum actually work? And of course, digging deeper into their evolutionary relationships using genomics, they offer a window into basic cell biology and evolution.
Right. So even if they aren't causing major human diseases based on what we've covered,
Correct. The text doesn't link them to major human or animal diseases.
their role as parasites, say, on algae, could have knock-on effects in aquatic systems, maybe even in aquaculture if important algae are being farmed.
Potentially, yes. If they parasitize a commercially important alga, or an alga that's a key primary producer in an ecosystem, they could have indirect impacts. Mostly, though, their importance lies in their contribution to overall biodiversity and the fundamental ecological processes they carry out. They're part of the planet's intricate machinery.
It really drives home that point about hidden complexity, doesn't it? This whole deep dive into Hyphochytriomycota, it's a reminder of just how much is going on at scales we don't normally see, and how science is constantly revising the picture, changing classifications, finding these unexpected evolutionary links like the one to algae. It's dynamic.
Absolutely. It shows that the tree of life isn't some fixed monument. It's a living hypothesis that we're constantly refining as we learn more.
Makes you wonder what other surprising connections or completely unknown groups are still out there, just waiting for us to look closely enough.
There's always more to discover. That's the exciting part.
Well, thank you for joining us on this deep dive today. We hope you feel a bit more clued into the world of Hyphochytriomycota and maybe just a little more curious about all the unseen life that surrounds us.